Hello, my name is Trinity Schuler, and today we'll be discussing how DNA makes us who we are. So first I want to talk a little bit about the structure of DNA, and what it is. So it is the hereditary material, so it's what you get from your ma and pa and your ancestors. There are 23 pairs of chromosomes in humans, and each species has a unique number of chromosomes, like dogs have 39 pairs and fruit flies have 4 pairs. The helical structure was discovered by James Watson and Francis Crick in 1953, which was based off of an X-ray diffraction image taken using X-ray crystallography, which was taken by Rosalind Franklin in the early 1950s. And the structure of DNA is it has a deoxyribose sugar phosphate backbone, and it has 4 bases, nitrogen bases, adenine, thymine, guanine, and cytosine. So how does DNA make us who we are? Well, there are really 3 parts. There's the evolution part, where DNA differences between species and ancestors make us who we are. The physical part, how DNA codes for proteins and how those proteins affect our appearance. And then the behavior part, suggesting our temperament and personality is partially determined by genes. So first, let's get into evolution. So it's going to have like a turn and talk situation, but since we don't have a turn and talk situation anymore, I'm just going to talk. So what is evolution? Well, in my bio textbook, it is defined as a descent with modifications. The idea that living species are descendants of ancestral species that were different from the present day ones. Basically, we all came from something. And how does DNA affect or contribute to evolution? Well, it creates genetic differences between parent and offspring, whether they're better or worse. So common ancestry, one species is the ancestor of 2 or more species. We all have an ancestor that gave rise to many life forms through speciation. Speciation is an evolutionary process in which one species splits into 2 or more species. So common ancestry deals more with DNA similarities, while speciation deals more with DNA differences, and we can prove speciation and common ancestry by looking at the genome of different species and comparing the similarities and differences. Let's take a look at a karyotype of a human and one of a chimpanzee. So a karyotype is just a picture of chromosomes, which are coiled strands of DNA wrapped around histone proteins. And as you can see, humans have 23 pairs of chromosomes, and then chimpanzees have 24 pairs of chromosomes. And they look pretty similar, except for chromosome 2. We have one long strand of chromosome 2, and theirs is split into 2 different chromosomes. But if we take a closer look at the components of the number 2 chromosome side by side, we can see there was a fusion of the chimpanzee chromosomes at the 113,602,928th base from the end. Well, we really can't see all 113 million bases, but we can see the locations in our chromosome that are almost exactly the same in chimps, suggesting humans stem from a shared ancestor. DNA similarities help us to predict common ancestors, while DNA differences help to predict how and where the human genome came to be unique. But how did modern humans come to be who they are today? Well, the final split from ancestors of chimps was about 7 million to 5 million years ago, and Neanderthals began evolving in Europe around 400,000 years ago and likely gave rise to modern humans by interbreeding with other ancient humans. But between the final split and the start of evolution in Neanderthals, we don't really know much. The missing links are known to David Reich as humanity's ghosts. These are the people we don't have enough information about to make a definite conclusion about their role in genetic history. We really only have concrete evidence for the last 50,000 years. And even then, there are still some discrepancies. Mutations in the genome can reveal secrets in past and present populations, but they also occur at the individual level. Physical talk. So what do you know about DNA transcription and translation? Well, if we remember the structure of DNA, it is comprised of nitrogenous bases, adenine, thymine, cytosine, and guanine. And stretches of these base pairings are called genes. And genes hold the instructions for protein synthesis. So DNA is first translated into RNA, the RNA is read by a ribosome, and a polypeptide or protein is synthesized. This is the basic process of gene expression. And what is the difference between genotype and phenotype? Well, the genotype is what you are born with, the genes that are in your DNA, and a phenotype is the expression of the gene. So an example of this would be hair color. Your genotype says that your hair should be brown, but you think it should be pink. So here is a little diagram of how DNA or genes are translated. RNA polymerase binds to the promoter region in the gene and unwinds as it transcribes an RNA molecule, the long red strand you see. Once RNA polymerase reaches the terminator region of the gene, the RNA strand is released and goes to a ribosome. The ribosome then reads the RNA molecule and a protein is transcribed. Once the chain of RNA is read completely, the protein is released from the ribosome and is transported to wherever it needs to go. Each gene or a group of genes have a specific code for a specific protein, so all proteins have a specific function. So examples of groups of genes coding for specific proteins are the keratin genes. These genes code for keratin proteins, a protein that make up your hair and nails. There are also the genes for eye color that code for proteins that help make melanin, the pigment that colors your eyes and skin and hair. These genes have base sequences that are specific to each individual. For example, I have blue eyes, and my friend Abby has brown eyes, which tells me the variation of the OCA2 gene, my variation of the OCA2 gene, which is known to produce a functional protein that helps create melanin, does not create as much of this functional protein in my eyes as does Abby's eyes, which makes me have less melanin in my eyes, leading to my blue eye color. Ultimately, it's the differences in our DNA that make each individual unique. We all have our own genetic makeup that sets us apart from others. And some DNA differences are noticeable, again, like your eye and hair color, but others are not so apparent, like that of your behavior and temperament. Behavior. So does DNA influence our behavior or not? Well, I don't think it's really so crazy and radical that it does. But if it doesn't, then what does? Our experiences. It's our experiences that influence our personality and behavior. So this section is pretty ambiguous. There's still a lot of research being done on it, so I apologize if something isn't very clear or if it seems like I'm off topic or jumping around. So, you know how I told you DNA codes for proteins and that's how genes are expressed? Well, that only applies to 2% of our DNA. The rest is what genetics call junk DNA. Junk DNA is currently known to play a role in determining a personality. How exactly it does it, they're not totally sure. It may influence gene expression, but there's still too little evidence to be sure. However, if we sequence the entire genome of many people, we can infer the effect of junk DNA. The first human genome was sequenced in 2004, and from there, scientists began to sequence thousands of genomes to compare them all, and in this comparison stage, they found millions of single nucleotide polymorphisms, or SNPs, changes in one of the 3 billion rungs of DNA. It is thought that the more SNPs someone has compared to someone else, the more genetically different they are, and the more different they'll be in personality. But what SNPs actually affect our personality? Short answer is, there is no short answer. There are no single SNPs that cause a psychological disorder or cause someone to be extroverted or introverted. Rather, everybody is on a spectrum. On the spectrum, there are extremes. So to the extreme end of the depression scale. But they actually don't really know what the other end would be. They don't know if it's extreme happiness or extreme, quote-unquote, normalness. But they can use SNPs to pinpoint how close you are to the extreme end of depression. But some of you may be thinking, but I thought the environment and our experiences was the main influence on psychological disorders in our personality. Well, you're not wrong. Our experiences do have an effect on our personality and our mental disorders, but the environment only partly influences us. It's 50% your DNA and 50% your experiences that influence who you are. This was proven through adoption and twin studies, which are used to determine whether there is any statistically significant evidence for genetic influence. So an adoption study is when a group of adopted children is studied through observation and tests to determine the personality and compare it to that of their adopted parents and their biological parents, like that of the Colorado Adoption Project. So if you're on the nurture side of the debate, you would expect a child to resemble the personality of the adopted parent. But if you were on the nature side of the debate, you would expect them to resemble the biological parent. Twin studies are similar, but they study twins. So they evaluate fraternal and identical twins and compare how alike fraternal twins are to each other and their parents, and how alike identical twins are to each other and their parents. So in the CAP and TED studies, they found that the child was more identical to the biological parent to an extent. In the TED studies, they found that identical twins to be more alike than fraternal twins to an extent. An adopted child can only be so much like their biological parent due to their genes they share and due to their non-shared environments. Identical twins may be about 100% genetically identical, but they still don't share the exact same environment. And so they found substantial heritability in childhood for lack of empathy and disregard for others, and they also found correlations in weight. I thought this was pretty interesting. They found that the style of parenting has little to no effect on the child, and it's all up to the non-shared environment. So we are genetically predisposed to react to certain environments, and it's those genetic reactions to non-shared environments that make siblings and parents different in your personality. Specifically, there are 3 types of gene-environment correlations: passive, active, evocative, and active. So passive has like an ambiguous correlation, so your environment is the result of your genetics. Evocative, your heritable behavior evokes an environmental response. And then active, one factor influences a preference for another. So you may be extroverted, so you want to go out and talk to people, but if you were introverted, you would want to stay there, and that would be a result of your genetics. So I know this was a lot of information, so we'll go over this kind of quickly. So evolution, our DNA, is a result of millions of years of branching off due to changes in nucleotide sequence. Physical. Genes code for proteins, proteins express genes, and genes and proteins determine how we look. Behavior, our temperament and personality is a result of genes and the environment, and it can be proven through twin and adoption studies. But ultimately, it's the differences in our DNA that make us who we are. It's what make us unique. Well, here's my Works Cited page. I thank you so much for enjoying my presentation, even though it was a little bit over time, and enjoy the rest of your day.